Today we're talking about a second macromolecule or large molecule called lipids. Lipids are also known as fats, but the word that you're going to see most often is the word lipid. So we need to know what it's made of, what we call them, and what their main jobs are. Lipids are made of carbon, hydrogen, and oxygen, similar to carbohydrates. But we're going to see they're arranged very differently. The main oxygens are actually going to be found here in the glycerol. So we want to know that this is kind of the backbone of every single lipid, is this 3-carbon glycerol. And attached to this glycerol are going to be hydroxyl groups, OH groups. And as we remove an OH group, we're actually able to then bind to the glycerol a fatty acid. Now, one thing to really pay attention to is that a fatty acid is mostly hydrocarbon. So mostly carbons and hydrogens. This means it's going to be very non-polar. Because it is very non-polar, it does not mix with water. And so we're actually going to call them hydrophobic or fearing water. So when we build a lipid, we have 3 places to put fatty acids. That is our backbone. We're limited here. It's not an unlimited polymer like carbohydrates are. It's a very limited polymer. We can only put 3 fatty acids, we call them tails, on there. We are still doing dehydration synthesis, so we're still removing water to build this macromolecule, but we are limited. So 3-carbon backbone, remove water every time we add a tail, we end up with this E shape. And so we do want to notice the differences in the shape between carbs and lipids. They're both made of CH and O, but they're arranged into a basic monomer. The basic monomer is going to be called a triglyceride, glycerol, glyceride, and then tri for the 3 fatty acids. But we're also going to see the suffix lipids. We'll see glycolipids, we'll see phospholipids. We're also going to see alls. So sterols or steroids. We're also cholesterol. We'll see a lot of cholesterol. So some alls are examples. The main job of lipids are actually very similar to the main jobs of carbs. They serve as energy sources. All of these carbon-hydrogen bonds are potential energy. So there's a lot of energy in lipid, but it's mainly going to be more long-term energy. It takes a lot longer to break down all these bonds. The other main job that we're going to see is structural. And if you remember, phospholipids are very, very important structures to the cell. We'll talk about them in just a second. So we need to know they're made of CH and O in a basic unit called a triglyceride. We're limited by the 3 carbons in the backbone for places to bind. And these tails, these ends, are going to be very, very non-polar, not attracted to water, very equally shared electrons. And so we're going to name our lipids a little bit based on their structure. We have saturated lipids and unsaturated lipids. Saturated, the term just really comes from the idea that you are saturated with hydrogens. And so this example here is a good example. Everywhere there's a carbon needing a bond, it's filled with a hydrogen. So there are no carbon-to-carbon double bonds, no double bonds. And so what sort of properties do saturated lipids have? Saturated lipids are going to stack very easily. And so this actually means that at room temperature, they're going to be solid. And so because these are going to be saturated and stacking and solid, these are usually the lipids lipids that are found in animal fats. So butter, the fat that's in butter is usually saturated fat. When we look at fats from other animals, like when you cook meat and the fat comes out, when you let the grease come to room temperature, it becomes a solid. Unsaturated fats do have carbon-to-carbon bonds. We could have one carbon-to-carbon bond, or we can have many. So monounsaturated and polyunsaturated are things you might see on the back of food labels. Now, sometimes what we'll notice is that foods can actually be altered. We can chemically alter the type of bond. But if we look at it in nature, unsaturated fats are going to mostly be your oils that are liquid at room temperature. Now, why would one of these fats be solid at room temperature and this one be liquid? These carbon-to-carbon double bonds actually form what's known as a kink. You can see it really clearly here in this picture. The carbons don't line up exactly in a straight line, and so they actually kink. And because they kink, they cannot lay flat, and this makes them more what we call fluid. They don't stick together, they're more fluid, and this is actually going to become very important in the cell membrane. We want the cell membrane to be a fluid mosaic model. We'll talk about that more in the next unit. But this ability to change fats from saturated to unsaturated is actually something that our cells do. And it just has to do with the bonding patterns. However, we know that bonding patterns influence behavior. So if I change the types of bonds, I'm going to change the behavior of this molecule. So this is your basic introduction to the structure of a lipid. But what we actually want to pay most attention to are a few specific lipids. First is the phospholipid. This is probably the #1 lipid to really, really understand. We see the suffix lipid, but then we see phospho, and we talked about these functional groups, phosphos, where we have the P attached to the O's, and some of the O's are charged. So lipids themselves are usually, we said, very nonpolar. Lots of carbons and hydrogens, equal sharing of electron. Even carbon-to-carbon double bonds are pretty nonpolar. So lipids are usually nonpolar. But a phospholipid has this charged phosphate group on one of the glycerols. So this is a great diagram. Your glycerol is actually turned a little bit different, so it's harder to see the E shape. Here are the 3 carbons of the glycerol. And we've got 2 fatty acids attached. Notice one of them is unsaturated. One of them is saturated. This one on the left. And the one in the middle is unsaturated. It has a double bond. That's why we see the kink. But the third space on the glycerol is actually taken up by a phosphate and a choline. You don't really need to know the choline. We pay attention to the phosphate. What this does is actually makes the phospholipid, this is one of my favorite bio terms, amphipathic. Amphi actually has to do with the word both. So instead of being all nonpolar or very polar, this molecule has a nonpolar region and a polar region. So therefore we call it amphipathic. The nonpolar region is what we call hydrophobic. Phobia, fear, hydrophobic. Oil doesn't mix with water. We know that. Fat doesn't mix with water. This phosphate group is charged. It's what we call hydrophilic. It likes water. It's attracted to the charge of water. And so this molecule is called amphipathic because the fatty acid tails make it nonpolar and hydrophobic. The phosphate group makes it polar and hydrophilic, so it actually has kind of 2 properties. And the reason this becomes so important is that these phospholipids, if you remember, orient to form a cell membrane. Now the cell membrane is actually a bilayer of phospholipids. So 2 layers of these lipids. And if you can remember, I was talking about the importance of water, and life is really surrounded by water. Both the outside, extracellular, and the inside, the cytoplasm, are made mostly of water. So these phospholipids, the phosphate heads, face the water. They're hydrophilic. The fatty acid tails that are hydrophobic orient towards the inside. And so this bilayer is going to face water with the phosphate heads, and the tails, what we call them the tails, are going to face away from the water. What this makes is a barrier, so that it is hard for things to get into the cell. This is actually what makes a cell exist, because it separates the outside from the inside. It makes a compartment for the cell to have. And this bilayer is chemically the reason that we can have a cell. Remembering that every cell has a cell membrane versus certain cells that only have cell walls. We'll talk about which cells have cell walls again later in the year. But it all has to do with the properties of bonding that create this molecule, phospholipid, and the properties of this molecule that create the cell membrane. What we'll talk about even more later in the year is all these other things that are in the cell membrane: proteins, glycolipids, glycoproteins. We are going to talk, though, a little bit about cholesterol. So cholesterol is actually an example of a steroid. When you think of steroids, you might think of people who are taking steroids to make their muscles bigger. So a lot of people think of steroids as muscle builders. But steroids are actually a form of a hormone. And all that a hormone does is communicate. And what these steroids are able to do is they're actually able to enter the cell. Because they are small and slightly polar and nonpolar, see this hydroxyl group? Slightly polar. The rest of these carbons and hydrogens, slightly nonpolar. Another amphipathic molecule. And because it's so small, it's actually able to fit right into the cell membrane. So steroids are hormones that can actually get into the cell. Other hormones are just going to have to attach to the protein and start a different way of sending a signal. But what we want to know is that there's a second class or set of very important lipids that have to do with the cell. The cholesterol itself is going to help with fluidity. It's going to help kind of break up those phospholipids and make the cell membrane more loose, more fluid. It'll be really important when it gets cold and the molecules stop moving. But then we've also got our other hormones like estradiol, testosterone, other steroids that are going to act as entering the cell and sending a signal. So cholesterol is an important one for fluidity. Other steroids are going to help send signals. So here are your questions that you need to be able to look at. What do carbs and lipids have in common? Of course, how are they different? What is important about the glycerol molecule, that backbone? What are examples of how lipid structure determines the function? And how are lipids an important part of regulation? If I need to control, that's kind of the other word, if I need to control lipids and control things in the cell, how do I do that? And that's lipids.